Hello, I'm Joni Rotten, and I have another Rotten research paper that I want to share. This one is from Wiley Online Library. It is an Access article, and it just came out on the 13th of February of this year, 2024. And it's titled The Last Stage of Development, the Restructuring and Plasticity of the Cortex During Adolescence, Especially at Puberty. So if you're following along, you will see why this is an interesting paper today. I'm going to struggle with a lot of these words, but I'm going to do my very best. So the abstract reads, there is considerable evidence of reorganization in the prefrontal cortex during adolescence in humans, as well as in rodents, where the cellular basis can be explored. Studies from my laboratory in the rat medial prefrontal cortex are reviewed here. In general, growth predominates before puberty. Pruning mainly occurs at puberty and after, with decreases in the number of synapses, dendrites, and neurons. The perineuronal nets, extracellular structures that control plasticity, are pruned peripubertally. It's just so many. Only in female rats. And I'm finding this to be extremely interesting. What I am reading here lately is going along with everything that is going on. Stay tuned because I have a lot more, almost more than I can even have time to share that I'm finding about female reproduction and adolescence. So keep a watch. So subscribe, like, and share my videos. I'll have more. Which may further open the adolescent prefrontal cortex to environmental influences. This is supported by recent evidence that exposure to just mild stress early but not late in adolescent decreases pre-pulse inhibition. And we'll get into what that is as we read. Additionally, exposure to methamphetamine in females early in adolescence increases the number of a major class of inhibitory interneurons, I'm not sure how to say this, but some other kind of neuron, while the opposite occurs later in adolescence. In females, even estrogen receptor beta mRNA decreases at puberty in the prefrontal cortex. Interestingly, rats of both sexes perform better after puberty on a test of cognitive flexibility in the water maze. Thus, evidence is accruing that adolescence is not a single entity, but rather an ongoing set of processes, and the environmental effects will differ depending on timing and sex. This is so fascinating. So we're going to kind of skip through because it is kind of scientific-y, but I'm going to read at least the first paragraph of each of these little sections so you get an idea of what this is researching. So, our introduction. There was an indication that the human cerebral cortex changes during adolescence as early as 1991 in an article. Subsequently, the papers by a couple of people further publicized that the cortical gray matter decreases in volume across adolescence. Confirmation that cognitive processes also mature during human adolescence complicated the neural data. The loss of volume during adolescence has led to the notion that it is only pruning that is occurring during this time. While pruning of many neural elements does predominate, growth also takes place, so that the overall picture of gross size changes is too much of a composite to capture the structural or reorganization at the cellular level. This means that studying rodent models is essential. The current review with summarized changes in the rodent, especially rat prefrontal cortex during adolescence, what is known about hormonal influences, and how the timing of environmental influences during adolescence can change the outcome since the reorganization is ongoing. The review is certainly not exhaustive, but rather is centered on the work from the author's laboratory, all of which is in both sexes. So let's talk about what's happening during the children. So when children are young, pre-puberty, their growth is what's mainly focused on. When you reach puberty, you start having the let me find the right word here, the pruning taking place. So pruning is where the mind and is getting rid of things that or reorganizing. It's pruning itself for the next phase of development, much like we use in, the butterfly and the cocooning. It's a cocooning stage. So that is kind of what they're talking about, that pre-adolescence or pre-puberty, the physical body is the main focus, and during puberty, it is the pruning that is the main focus. Although some growing is done, the main objective is the pruning. So the opposite of before that. So they've got a lot of figures and things. So part 2 says that adolescence and puberty, the link to this is in the description if you'd like to read the entire thing, or disagree with my interpretation and or want to share it yourself, or tell me where I was wrong or what I missed. The exact timing of adolescence is arbitrary, and there is no single metric, either behavioral or neural, that defines it. In her seminal review, Spear estimates, this is from 2000, that dates for rat adolescence from postnatal day, so 28 to 42 days. So you have a bit of an area that you can, but you can't say this exactly the same way that most, every child, most children, the average child is going to walk at 12 months, but that does not mean that they're going to walk on their 12-month birthday or their 1-year birthday, and or pre or post. As long as it's within a healthy range, it's okay, and it's a normal behavior. So that is what they're saying here, that as long as it's within the range of the years of puberty, which can be about 5 years or so, less than, then you've got them covered. So if you can hit them during that stage there, you've got about a 5-year window that you can hit an adolescent and take advantage of the synapses that are being pruned. So this is mainly based on changes in behavior. Considerable work has occurred since its original formulation. Here, I would like to propose that the rise of hormones surrounding puberty is the principal marker that extends adolescence beyond P42, especially for male rats. This is based on neural and to a lesser degree behavioral data. The rat is an excellent model for detecting the onset of puberty, since the hormonal rise is very abrupt and results in exterior morphological changes. For example, the vaginal opening and the preputial separation. Preputial? That can be readily observed. So it's a quick change in rats. You have a better window, whereas in a larger animal, it would be more difficult. There also is a reliable separation between the timing of puberty in females and males, which allows for a dissociation between age and pubertal status. This contrast with adolescence in human, which extends over years. Nevertheless, there's evidence of some neural changes related to puberty in human adolescence. It should be noted that mice, on the other hand, reach puberty at variable times, depending in part on strain and housing, with some overlap of the age. Okay, so there's the figures for that. So then the next part of this is the adolescent pruning and growth. The decrease in volume of the prefrontal cortex during adolescence in humans implies that there is pruning. This has, in fact, been documented in post-mortem human tissue, despite its limitations of availability. Although some guy in 1979 had only one adolescent brain in the sample, his data indicated that there was a loss of synaptic density as well as the number of synapses per neuron between childhood and adulthood in the prefrontal cortex. They found in 2011 that dendritic spine density on both layers 3 and 5 of the Gogler-stained human prefrontal cortex decreased between childhood and the third decade. In addition, in 2007, using a marker of synaptic junctions, synaptixin showed that there was a decrease in the prefrontal cortex between 6 and 10 and between 16 and 20 years of age. So you've got a 6-year kind of window going on there. Thus, it is clear that synapses are pruned between childhood and adulthood in the human prefrontal cortex. However, these studies do not have a significant number of subjects to examine sex differences, and they certainly do not speak to the potential role of puberty, but they are noting them. So let's keep going. Again, these are so interesting. So here you have basically they're just talking about all of their differences here, boys, girls, etc. And so you've got some figures. And so not everything shows evidence of pruning during adolescence. We found that fibers containing tyrosine, hydrolax, the rate-limiting enzyme for dopamine synthesis, grow from P25 through adulthood in rats of both sexes. And it goes on to talk more about some of those things. Okay, let me go down to, there is another. So here's the number 4, the role of puberty. In both sexes show an overall loss of dendritic spines and neurons in the rat MPFC, but the losses in males are not as large as in the females. In the visual cortex, we have removed the testes and ovaries before puberty, P20, and found that ovary-ectomized females had more neurons, which were the same number as intact, and I'm not sure, gonectomized males. I'm not sure how to say those, but you get the idea. The ovaries are removed, it's implicating ovarian hormones as the cause of neural pruning. So that's what that indicates. So something about the ovaries or the ovarian hormones might be why girls are having this this larger portion, right? Not as large as in females. Likewise, removed the testes and ovaries before puberty and found that the MPFC, that the number of neurons in the female was higher, and again, there was no effect in males. So you can see that there's their little, you can see a significant jump in the girls and not so much difference in the boys. So let's go down to the next section. And so then it goes on to talk about this. We have a series of studies that indicate an important role for estrogen in the reorganization of the MPFC at puberty. And so it is the receptor that is present during early cortical development. Okay, and more sciencey papers. I'll get down here. You can see some more figures. Okay, so these results indicate that outside of the hypothalamus, the MPFC may be one of the neural regions that are exquisitely sensitive to the rise of estrogen at puberty. And that comes from the motor cortex. A nearby frontal area is especially noticed, the difference. How all this correlates with behavioral changes is difficult to establish. Many studies have compared behavioral performance between adolescents and adults, including those from our laboratory, and differences occur between adolescents and adults on most tasks. One shortcoming of many studies of adolescents is that they cannot easily parse the effect of puberty because training and testing may take too many days. One task that can be used is the water maze. While this is not a difficult task for rats requiring little MPFC input, changing the position of the platform after training requires cognitive flexibility, which taps the MPFC function. We trained adolescent rats in both sexes on the water maze. Some groups were pre-pubertal by 2 to 5 days, and some were post-pubertal by 2 days, and also at P60. There were no effects of puberty on standard training. When the location of the platform was moved on the second day, the post-pubertal rats adjusted to the new location, shorter path length, better than the pre-pubertal rats. Interestingly, the females that were post-pubertal by 2 days were younger than the pre-pubertal males. So it is the pubertal status rather than age that influences cognitive flexibility in performance. I think that is very interesting. So going along with what we've kind of already been known and told, that girls mature much faster than boys, and this is part of that. So the fifth thing they have here is timing within adolescence matters. It matters a lot. So when we have a direct generation of children on the planet that are in their puberty time frame, so that would be all the Gen Zers during the COVID, or say the millennials during 9/11, we are having a timing with adolescence. It creates an outcome in the future. If you've been following along with Joni Rotten, you'll know that I've been talking about that for a very long time. Although I get dismissed, 9/11's not about the children. No, of course not, but it is about the children. It had an effect on them. COVID had an effect, not on the babies, on the puberty children, the ones that are about to become the next adults. As the above experiment indicates, pubertal timing may be more important than age during adolescence. Outside influences can have disproportionate effects when cellular changes are occurring. Another example from our lab is the effect of a mild stressor, one like isolation, followed by 1 hour of isolation, followed by 1 hour of restraint during the dark cycle. Just that. A mild stress, being isolated and held back for only 1 hour. Now, can you imagine if it was for 3 years? This was applied to 7 days, either across puberty or after puberty, with different timing for each sex. And you can see that in this figure here. A battery of tests were run in adulthood, and a principal component analysis revealed that 1 of 4 factors showed a significant effect of treatment. This factor loaded on the pre-pulse inhibition, or the PPI task, which measures sensory motor gating, which is the reduction of a startle to a loud noise. Thank you. Okay, I'll be out there in just a second. Very good. That's very good. I got a picture from my wee one. So let me reread this, because this is a very important, this is the meat of it. This is the important part to note. It says this factor loaded on the pre-pulse inhibition task, which measures sensory motor gating. And what is sensory motor gating? Okay, so sensory motor gating it refers to the regulation of sensory information as it is transmitted to the motor output systems. Sensory motor gating refers to the regulation of sensory information. So what sensory information is coming in at me? What environmental factors, what stresses physically or mentally are coming into me? It's the regulation of this sensory information as it is transmitted to motor output systems. So your fight, flight, and freeze systems. This factor loaded, I'm going to read it again and continue. This factor loaded on the pre-pulse inhibition task, which measures sensory motor gating, the reduction of a startle to a loud noise when there is a weaker warning noise. That's what they're doing. So, you know, if you were to have a bang and you were to, ooh, what was that? You know, reaction. What they're finding is that they can reduce that reaction. The reduction depends on inhibition from the MPFC. That's from 1994. They've been at this a minute if you'll notice the dates. We found that the peripubertal stress groups of both sexes have impaired sensory motor gating, remember what that is, compared to non-stress controls of the post-pubertal stress groups. This occurs in parallel to in both sexes, in spite of a main effect of sex, such that females have lower PPI scores than males. It is also notable that the age of the post-pubertal females overlaps the age of the peripubertal males. And again, they're ahead of the game. Another example of timing of environmental influences not being constant during adolescence comes from work that is currently unpublished, I'd like to note. In collaboration with a colleague, we injected rats of both sexes with methamphetamine for 9 days, either in early adolescence across female puberty or in late adolescence across male puberty. 24 hours later, their brains were harvested. In females, but not males, meth increased the number of PNNs across ages. Increasingly, the number of PV neurons also changed in females during early adolescence. Interestingly, the number of PV neurons also changed in females. During early adolescence, meth exposure increased the number of neurons detectably expressing PV. In contrast, in late adolescence, the number of PV neurons was lower following meth exposure. PV neurons in males were unaffected by meth exposure. Again, some more people with an unpublished data, so it is not published. Why is it not published? Ask yourself that. This study shows, again, that the effects of environment can change depending on the timing within adolescence, especially in females. Whenever they don't publish data, the data is available, but it is not made public. Unpublished is unpublic. And here are the conclusions. Consider pruning occurs in the rat MPFC of both sexes, particularly in females. However, there is also evidence of growth and incoming axons containing TH and increases in dendrites, dendritic spines, and synapses early in adolescence before puberty. Most of the pruning occurs after puberty, again, especially in females. This indicates that adolescence is not a unified entity, but rather varies with sex and proximity to puberty, which can change the type of influence that the environment can exert. In both the literature from non-human and human subjects, adolescence has been acknowledged as a developmental period over 20 years ago. Now we need to understand the behavioral and cellular complexity during this developmental period because we can manipulate it. And so there is some more figures. And our acknowledgments, we would like to thank all of the co-authors who have worked in my laboratory on pieces of this puzzle, grants from the NSF and various NIH institutes have supported these studies. Way to go, guys. And ongoing work is currently supported by, and it does give me a number I can go look at. A conflict of interest? No, we have no conflict of interest. Our open research tab says that no new data has been generated for this review. The ongoing unpublished data will be available after it is part of a peer-reviewed publication, once they decide to share it. And then we have a few references, I believe, here. Yes. So you can see that they've been at this for a hot minute. PubMed. Adolescence. Stress during, but not after. Look at that. So anyway, that is it for this paper. I do find this interesting. I have a lot more things. Just think about what is being talked about serial wise, and that endocrinonic. Is that an endo? I can't say it now. System that I've been reading lots of papers about it, but I can't. Endocrinatic system. A lot of papers there. What is going on? I just today, like literally just this morning, I've been doing all this research about, precocious puberty. A lecture comes across from one of the universities or research things that is describing the evolution of the ovaries. So it's being discussed. That was just uploaded today. So it is hot topic. This right here, puberty, reproduction, infertility, eat your cereal, poisons, PFAS, environmental factors, all over that paper as well. Environmental factors, stresses, foods, air, water, all of these things add to the outcome of that child into an adult. And notice that it said behavioral changes. And there's more research that talks about that. Anyway, I hope you find this stuff as interesting as me.